on animal model of Alzheimer's disease, the preclinical approach on the cognitive dysfunction. So this research cannot be done without the help of ARAS, because the source that we got for the generating the data in this research is basically from ARAS, and they are the one that's supplying the animals and providing all the food and the beddings for the research. So during this presentation, I will cover on the dementia and Alzheimer's disease. Then I will go to the preclinical studies using animal model, chronic cerebral hypoperfusion, and then what is the task that we are going to use in animal model, especially to look at the cognitive function. So there are several behavior tasks that I will cover, and then at the end of the presentation, I will cover on the neurotransmission between the neurons. So how this transmission between the neurons help in retention memory. As you can see, dementia. 2 out of 3 people globally believe that there is little or no understanding of dementia in their country. So very little people knows about dementia. About 152 million people living with dementia by 2050, almost 68% in the low and the middle income countries. But most people doesn't understand about this symptom, because the symptom is associated with a decline in memory, reasoning, and thinking skills. It's not a normal part of aging, even though when you are getting aging, you will develop this cognitive impairment. However, this is not the normal aging. It's caused by the damage to the brain cells that affect their ability to communicate thinking, behavior, and feelings. So dementia has several types of dementia. So there are 5 types of dementia that most people doesn't know about this. The first and the most common dementia is Alzheimer's disease. About 60 to 80% of the cases of dementia related with the Alzheimer's disease, followed by the vascular dementia. Vascular dementia is the second most common dementia due to the complication of vascular disease, such as hypertension, stroke, and diabetes. And then we have this Lewy body dementia. It's about 15%. It can cause psychosis, such as hallucination, for those people who are having dementia. And then we have this Parkinson's disease. At the end of the stage of the Parkinson's disease, about less than 5% of people will develop dementia. And the last one is actually on the frontotemporal dementia, whereby there is a development of abnormal protein at the frontal and temporal lobe that cause the changes of the personality, emotion, and the behavior. And at the end, you will have a communication difficulty. Some of this dementia can be a mixed dementia, whereby you have more than one dementia. So as I mentioned earlier, Alzheimer's disease is one of the 60 to 80% cause dementia. So this Alzheimer's disease is actually a progressive neurodegenerative disease that damages healthy cells in the brain, cause cognitive impairment and functional disability. Most people doesn't know about this because they thought that Alzheimer's disease is one type of the different disease of the dementia. As you can see, in Europe, about 10 million people are having this dementia, and it is expected to increase to 14 million by 2030 due to the aging population. And this dementia has been known as the 5th leading cause of disease by World Health Organization. The symptoms of dementia can be categorized few, like difficulty in everyday tasks, confusion in familiar environments, difficulty with words and numbers, memory loss, and changes in mood and behavior. Please don't mix up with the aging. What happened in the Alzheimer disease? As you can see, Alzheimer disease has been caused by the beta amyloid protein. This beta amyloid protein produced normally in the brain, in a healthy brain. However, it will be broken down and removed from the neurons. In the Alzheimer disease patient, these proteins build up and then it will form a cluster and phosphorylated the tau, and at the end it will form the neurofibrillary tangles. What happens is actually it will block the synaptic transmission and prevent the neurotransmission from one neuron to another neuron. So at the end, the neuron will cause degeneration and cause a neuronal death. So as you can see, in Alzheimer's disease, the size of the brain will be shrink and when you have a severe Alzheimer's disease. So this pathological stage is one of the cause of the Alzheimer's disease. You can see the difference is actually on the size of the brain. As you can see here, one day you may develop a loss of password. You cannot remember even a single simple word. So as I mentioned earlier, Alzheimer's disease can be categorized into several stages. The first one, known as the mild cognitive impairment. This will take about 7 years, and the disease begins in the medial temporal lobe, whereby the symptom cause the short-term memory loss. In the mild Alzheimer's disease, it will take about 2 years, and the disease spread to the lateral temporal and parietal lobe, and you will have the problem like reading, poor object recognition, and poor direction sense. And then, in a moderate Alzheimer, it will take about 2 years. The disease will be spread to the frontal lobe, and then you will form the symptoms like poor judgment, impulsivity, short attention, and for the severe Alzheimer, take about 3 years. The disease will spread to the occipital lobe, and you will have a visual with the problem. As you can see below here, the size of the brain also shrinks when you have the severe Alzheimer's disease, such as the shrinkage of the cortical, you will have enlarged ventricles, and also shrinkage of the hippocampus, the main region which involves in the cognitive function. So the continuum of Alzheimer's disease, this has been done by the Alzheimer Association based on the biomarker that can be produced to develop the stages of the Alzheimer's disease. As you can see here, we have this cognitive function and then throughout the years. So the stages start with the preclinical, whereby during this preclinical, you will develop asymptomatic, means that you don't have any symptoms, but you will develop a memory loss. But there is also some changes of the brain. And then we can detect this preclinical through the changes of the biomarker such as amyloid beta, tau, or through imaging. And then the next stage is the MCI, is a medial cognitive impairment, whereby you have a normal forgetfulness, which happen to have a deterioration of the thinking ability, unable to plan, unable to work independently. And then the last one is a dementia, where you will develop a clinical sign. As you can see in this graph, the aging and dementia are two different things. So aging will develop cognitive function, but a bit slow compared to the dementia. The cognitive function will develop very abruptly. So what are the symptoms of Alzheimer's disease? You will have memory loss, changes in mood, social isolation, poor judgment, wandering, problem with the words, misplacing things, hard to complete tasks, confusion with the time or place. All this can be prevented using the brain activation, exercise, diet, or drugs. So these are the drugs that have been used to treat Alzheimer's disease: cholinesterase inhibitor, NMDA antagonist, and either combination of both drugs. Cholinesterase inhibitor, the most common use are donepezil, rivastigmine, galantamine. All these drugs, cholinesterase inhibitor, normally used for the mild to moderate disease. For those who are having moderate to the severe, doctor will recommend you to take memantine, an NMDA antagonist. Or you can also take the combination of both drugs. However, this drug producing a lot of side effects, and you can't even use one. So maybe you have to use a combination of drugs. So what happened in the lab? We try to develop a new drug for improving memory in the Alzheimer's disease. To develop a new drug, you have to go through a few stages. The first one starts with the basic research, and then you're targeting the early validation, compound screening, proof of concept, or candidate selection. All these are categorized as a preclinical drug development before you go to the clinical development, which is phase 1, 2, and 3, before the drug can be approved by the FDA. So in the lab, what we are doing, we are developing a preclinical animal model known as a chronic cerebral hypoperfusion. So this model has been identified as a cerebral ischemia, as it also developed a vascular risk factor linked to the vascular dementia and Alzheimer's disease. As you can see in this model, what happened? We will occlude the common carotid artery of the main blood vessels going to the brain. So what happened? The oxygenated blood to the brain will be congested and resulting hypotension. You will develop a low blood flow to the brain, so sufficient to induce brain damage and lead to the cognitive impairment. So using this model, we can screen a lot of natural products with a therapeutic potential value in vascular disease. So the models that we are using is actually known as a permanent bilateral occlusion of the common carotid artery. So this model has been known to produce a lot of molecular mechanisms similar to the vascular dementia. As you can see, our review paper, the latest our review paper last year on the neuropathology of cerebral ischemia, you can see what happened in this animal model. So when you occlude or blockade the carotid artery, there will be low blood flow, reduce the oxygen and nutrients to the brain, and also reduce the ATP production. At the level of the neuron in the brain, they develop a glutamate cytotoxicity. What happened? The level of glutamate is very high in this. And also, it develops cholinergic dysfunction. So this is one of the mechanisms that's been used for developing a new drug, cholinesterase inhibitor. So in this molecular pathology, as you can see, when all this happens, it will develop increase in the ROS, reactive oxygen, and then you will activate the mitochondria damage, lead to the apoptosis or the cell death. In the same time, activation of the astrocyte or microglia will develop inflammatory response that leads to the cell death. And also, increase in the production of hydrogen peroxide can develop into the cytotoxic edema, which may have a problem with the blood-brain barrier destruction. So all these, the neuronal damage, white matter lesion, glia activation, and BBB destruction lead to the cognitive impairment. So this is what we are going to see in the animal model. So we have developed a model known as a chronic cerebral hypoperfusion or chronic cerebral hypoxemia. So we look at the cognitive function of this animal model. As you can see, this animal model doesn't develop any motor dysfunction. So it acts normally as a normal animal. And then for the escape latency, one of the activity or tasks that been used to test for the spatial memory. So as you can see, the escape latency in the animal model POCA increased, means that the animal has a difficulty in finding the stage in the Morris water maze. And in the passive avoidance task, as you can see, it takes a shortened period of time to go into the dark compartment. So what happened, I will show you later details on this method. And then the expression of the BDNF also decreased in the model, and it will increase slowly as the model produce a regeneration of the neuron. Here you can see at the hippocampus level, the pyramidal neuron showing a pycnotic nuclei due to the neuronal death. So as I mentioned here, one of the characteristics to develop a new drug for the Alzheimer disease by looking at the cholinergic system. Cholinergic system, the neurotransmitter known as acetylcholine. So acetylcholine is the neurotransmitter that play a role in learning and memory. However, in the Alzheimer disease, these acetylcholine are very low in the brain. So what happened? There are two enzymes that cause the breakdown of this acetylcholine, cholinesterase enzyme and also butyrylcholinesterase enzyme. So both of these enzymes will break down the acetylcholine into the choline and acetate, and then it will go uptake into the brain and form back acetylcholine in the presence of CAT, cholinesterase acetyltransferase. So what happens if you have less acetylcholine? You will develop cognitive impairment or memory loss. So it's very important to develop a drug which can inhibit this enzyme from breakdown the acetylcholine. So the first methods that we use in the lab to test any potential drug or any potential natural product that can be developed as a smart drug by looking at the in vitro assay, so known as the Ehrman cholinesterase assay. This normally performed in all plant samples to test for their cholinesterase activity inhibition. If the plant has a very high potential, 99% or 100% cholinesterase inhibition, so that is the best natural product that can be tested further in animal model. The best natural product that produce this high percentage inhibition must have a very low IC50. So from here, we test in animal model using several neurocognitive tasks. First of all, let me show you what is happening in the memory. So human memory are divided into three: sensory memory, short-term memory, or long-term memory. The sensory memory is the first memory that been received by our brain. It will stand about less than 1 second, followed by the short-term memory about less than 1 minute, and then if you are doing that multiple times, activated the brain multiple times, you will develop into the long-term memory, which can be kept in a lifetime. So as you can see here, the long-term memory can be divided into the conscious and unconscious, and also from the conscious, you will develop the declarative memory, whereby you will have episodic and semantic memory. For the unconscious is the procedural memory, whereby when you learn how to ride the bicycle, means that when you learn daily, it will develop into the long-term memory and it will develop into the procedural memory. Other than that, like experience or the fact is developed through the declarative memory. In the lab, these are the cognitive behavior tests that we are using. So we either looking at the spatial memory task, contextual memory, or working memory. One example of the spatial memory task is Morris water maze. So these are the tasks that we've been used in the lab to study the spatial memory. And then we have this contextual memory using the passive avoidance task, whereby you have 2 compartments, and in the dark compartment, the animal will be given current, a small electric shock. So the next day, during the retention time, the animal will remember and they will feel very hard to go into the dark compartment. So it will increase their escape latency. But those animals who cannot remember, they will go very fast into the dark compartment because they have a loss of memory. This passive avoidance task involves the region of the brain known as amygdala. In the working memory, we have several tasks like white or T maze, open field, and also object recognition tasks. So I will start with the spatial memory. These are the brain regions that involve in the spatial memory. The main common is actually hippocampus. There are also other regions like prefrontal cortex, entorhinal cortex, parietal cortex, and retrosplenial cortex. But the main regions involved in the spatial memory is hippocampus. So as I mentioned earlier, one of the tasks used is actually Morris water maze. These are related with the hippocampal spatial learning memory. So what happens, you will have a swimming pool whereby you will have a platform in one of the quadrants. So animal will be released into the swimming pool and it will swim to find the platform. So we will find the time taken for the rodent to swim to the platform to escape the water, because animals normally they don't like water. So they try to escape from the water by finding the platform. So the ability to identify a spatial location based on the visual cues placed around the chamber. And during the probe trial, whereby there will be no platform, so the animal will spend, if the animal remember the place, they will spend a lot of time in the platform region. As you can see, this is the data that we generate in the lab. So we try testing using the xanthone extract fraction from the Garcinia mangostana, which is known as a mangosteen. So we tested several fractions of the mangosteen, and also we used a compound of the mangosteen. As you can see, the blue color, you see when the animal swim and then they try to find the platform. As you can see, in the normal animals, they spend a lot of time in the quadrant. So the escape latency will be very short. So means that they find the platform very fast compared to the model, the animal model that we use, the POCA model. You can see there's a high increase in escape latency. So as you can see, when you treated the animal with the extract, the escape latency reduced. The best one is at the alpha mangosteen 15. So in the same time when you are testing using the probe trial, these animals spend more time in the target quadrant, almost similar as a normal rat. In the passive avoidance task, as I mentioned earlier, if the animal can remember, so they will take longer time to go into the dark compartment, because they remember on the other side it will be a electric shock. So as we tested using, this is the plant known as a Clitora teneti, or bunga telang in Malay, normally used in the production of the nasi kerabu. So it's a blue color. But here we use the root instead of the flower. As you can see, in our model, the POCA model, the animal is unable to remember. So it will be a very short time and then it will go into the dark compartment. As we increase the concentration of the extract, it starts to produce a memory. So you can see at the higher dose concentration, the step-through latency almost similar as a normal rat. In the automated open field, one of the tasks to look at anxiety or the activity, the motor activity. Using the alpha mangosteen, there is no significant changes in the motor function. So this task has been used to not only looking at the locomotor activity, the distance traveled, the speed, and also time spent in the center. If those animals spend a lot of time in the center, means that they are developing anxietic effect. So the next step is actually to look at the novel object recognition to assess the animal's innate ability to distinguish an old from the new object. This is to evaluate the recognition memory. Normally, normal rodents remember the old object and spend more time exploring the new object. So what we'll do here is to look at the parameters like time spent interacting with both objects. As you can see here, so during the training day, you have two similar objects, object 1 and object 2. During the testing day, you replace the object 2 with a new object known as a novel object. So we identify the interaction between both objects. As you can see, exploration time taken for the normal object, they normally spend more time in the novel, in the new object, but not for the transgenic animal. They spend almost similar time in both objects. And also the discrimination index for the transgenic is also reduced compared to the control. So this is actually the video to represent how the interaction of the rats to the familiar and novel objects. And these are the system that we use, like smart system, to identify the interaction between the objects. For the part 2, I will cover on the neuroplasticity. As you can see, this neuroplasticity is very important in the formation of the memories through the changes of the synaptic recording using the long-term potentiation, LTP. What is LTP? LTP is actually a single transmission between the two neurons after repeated stimulation. It's a very popular mechanism to explain the changes in the synaptic connection. So as you can see here, the mechanism involved in LTP by enhancing the neurotransmitter, activation of the AMPA, or changes in the number of synaptic spine contact. If more spine producing, so means that more memory is kept. So in the long-term potentiation, also known as the long-lasting enhancement of the synaptic transmission, can be induced by a brief or repetitive stimulation. So what happened here? This is at the initial state of the neuron. So you have a pre and a post. So when you give a small stimulation, it only happens to have a very little number of receptors. But if you keep repeating the stimulation every day, and then they will develop a long-term potentiation, whereby after 1 week later, you can see the number of the receptor increase. So more neurotransmitter can bind to the receptors, and it will increase the action potential. So this is what happened when you read during the exam. We normally read, if you want to remember the things, we read multiple times so that it will store in the memory as a long-term memory. So this is a very important because this is the model to investigate the cellular mechanism involved in the learning and offer an attractive hypothesis on how memories are formed. So as you can see here, what happened in a normal situation, you will only have a few number of the receptors at the post-dendritic cell. And then after you giving a lot of synaptic strength, long-term potentiation, the neuron will be activated, potentiated, we call it, and then you produce an increased number of the receptors so that more neurotransmitters can bind to these receptors. So what happened? This happened in the region of the brain known as the hippocampus here. So this actually photos taken from the rat animal. As you can see, the structures of the hippocampus, the memory starts from the perforan fiber pathway, pass through dentate gyrus, CA3, and to the CA1. So as you can see here, the region of the dentate gyrus doesn't have any NMDA receptors. However, at the CA1 and CA3, you can see the blue color, there's a NMDA associated neuron. So this is what we do in the lab from the technique that I learned from Canada. So I managed to set up another set of the lab in the CDR. So we are investigating the connectivity among the neurons, known as a neuroplasticity, which provide the mechanism for the brain to develop, change with experience, and encode long-term memory. Because you can ask the animals to stimulate its own brain. So what happened here, we are stimulating the brain to create a synaptic strength inside the brain. So in this in this animal, what we do is actually we are stimulating one region known as a CA3, and we are recording at the CA1 region. So the action potential, because there is a nerve, there is a neuron, afferent neurons that passing through, known as a shuffle collateral from the CA3 to the CA1. So these are actually the hippocampal circuit that happen in the brain. So as you can see, the yellow is actually how the memory is passing from one region to another region. So as I mentioned earlier, we are stimulating the sharper collateral, and then we are recording at the CA1 region. So these are the setup of the lab, setup of the surgery in the lab. So as you can see here, if you are giving more information, you are like stimulating the brain, developing a more action potential, and we can develop into the late LTP. In the early LTP, only develop in a small frame, means that you're giving a very small stimulation. But if you are giving high stimulation, you will develop a late LTP. So in the Alzheimer disease model, what happened? This is a normal healthy synapse between the connection between the neurons. When you develop the Alzheimer disease, you develop the alpha beta oligomers, known as I mentioned just now is a beta amyloid plaque. So what happened? This amyloid beta will cause the internalization of the receptor. So means that you will develop a less number of receptors on the synapse. So most of the synapse will be internalization inside the cell. So at the end, you will develop a thinning and loss of synapse. So the number of the receptor will be reduced, and the size of the neuron also reduced. And this will lead to the cognitive impairment. So in the lab, what we do, we're measuring the action potential by giving a different stimulus intensity. We want to see what happens at the action potential of the neuron on the postsynaptic. As you can see, when you increase the intensity stimulation, you will increase the action potential. However, when you see here, this is in a normal rat. In the POCA model, the animal model that we developed, you can see it's a very short, very low amplitude developed when you increase the stimulus and intensity. So this is what happened. You can say that this is what happened at this transmission neuron. The number of neurons, a lot of neuron death. That's the reason why it produces a less amplitude compared to the normal rat. However, when you give a strong stimulation, this is what we do, the recording of the LTP. We can see when we give a strong stimulation known as the theta burst stimulation, the normal will create a standard or maintain production of the amplitude. However, in the POCA model, it will drop. So means that it cannot stand longer, means that the memory is lost. However, when you give treatment with a natural product, as you can see, there's a increment of the amplitude, the action potential in the animal model compared to the treatment in animal model. In the same time, we're also looking at the pathological state of the neuron in the pyramidal neuron. As you can see, there's a lot of the hypnotic nuclei production at the pyramidal neuron in the hippocampus region. So it means that there's a lot of neuronal death in the POCA model. So as a discussion, in this animal model, we have shown that polytorial-alternative root extract can rescue the long-term impairment, and it also protect the synaptic function by improving the effect of the polytorial-alternative on memory and learning. So as a summary, polytorial-alternative has been shown to be a good model for the potential therapeutic treatment for the vascular dementia. I just want to highlight it to you. In our lab, we're also doing this technique known as a microdialysis. What happened in this microdialysis is we're collecting the brain sample in a real time. So we can measure the extracellular neurotransmitter release at a specific region of the brain by collecting the sample in every 20 minutes throughout the whole experiment. So Doing this, you can measure the changes of the neurotransmitter in real time, and you can analyze what is happening when you are giving the drug. So this is the best method to measure the neurotransmitter in the specific brain region. So as I mentioned earlier, the models that we use, the POCA model, has been used to develop a lot of smart drugs as a potential therapeutic strategy to prevent the progression of the cognitive deterioration in vascular dementia. And most of the drugs that we develop is based on the techniques that I mentioned earlier. So from here, that's how we develop into the preclinical studies. So as I want to end my presentation, I just want to highlight to you what you can do to prevent your dementia by watch your BMI, exercise regularly, intervene early diagnosis, eat healthy food, get an education on this disease, look after your heart, and limit the alcohol intake, and you will end up with a happy and healthy person. So, and I also want to introduce you that we are organizing another e-symposium in conjunction of the International Conference on Drug and Discovery Translational Medicine in 2020. So we will have a free webinar on the addiction, what we know and way forward on the 24th February. You can have the link for the registration. And I would like to thank for all my students and my collaborators, especially Professor Hans, that has been produced and has spent a lot of time with me to teach me how to do this synaptic plasticity, and also all my colleagues and my students that has been developed a lot of data, good data to be published in a very good journal. So I thank to all my collaborators and also ARAS for producing a good source of animal to have a very good data in our good journals. Thank you.
I didn't understand that.
Alhamdulillah, thank you very much, Associate Professor Dr. Zainal. It's a very, very interesting presentation. But I have to hold the session